So this topic is on organelles called mitochondria and chloroplasts, and they are associated with some energy metabolisms that we see within cells. And so we're going to look at learning outcomes for this section is to describe the structure of both mitochondria and chloroplasts. I'm going to recommend that you sketch and be able to recognize features for these organelles here and some of their basic parts that you see in there. So try to practice sketching them and relabeling them so that you learn them, because the structures become very important in future chapters when we talk about how these organelles function to generate and transform energy within the cell that the cell uses. So that's your first objective. Make sure you learn that in this chapter so that the future chapters become easier. Second objective is to compare the functions of each one. Mitochondria and chloroplasts are different, what they do, and then explain where these organelles came from. These organelles, their origins is not the endomembrane system. They have a different origin. So we just finished talking about endomembrane system. These are membrane-bound organelles, but we're going to see that there is evidence that suggests that they actually originated outside of other cells and then came to live inside of cells, a theory called endosymbiosis. And endosymbiosis, endo means within, and symbiosis means to live together. And we'll take a look at a little bit of that evidence that supports this theory. So we're going to look first at mitochondria. And mitochondria, a lot of times they're colored red. They kind of remind us of energy, hot, right, fire, that kind of stuff. But they're not always colored red. So I'm going to recommend that you be able to sketch. I'm drawing in red right now. But you should be able to sketch the basic structure that has an outer membrane and then an inner membrane. And these are phospholipid bilayer membranes. And the inner one is folded. And so my drawing is better than these because I sketched it, and then you can start labeling the outer membrane, inner membrane, and some of the important structures there. If I drew a chloroplast in red ink, you should be able to tell based on the characteristic structure of it, not the color of it. So be aware of that. On tests, they may not be colored red or green. So mitochondria are going to be found in all eukaryotes, and they are bounded by membranes. There's an outer membrane, which is that one there, and it's labeled right there. There's going to be the inner membrane space. So the inner membrane space is the space between the inner and the outer membrane. Inter means in between. And then there's going to be an inner membrane, which is what it's called, and it's going to have folds called crista. And crista is as plural. Crista would be singular, so you can see they're pointing at one fold and they only ended in an A. So the rules for singular and plural are a little bit different when they use the rules of grammar in Latin. And then the matrix is going to be the fluid-filled, gel-like fluid-filled inside the inner membrane. And that's going to be where a lot of the action takes place that we're going to learn in future chapters when we look at how mitochondria break down food to generate ATP. Now, I'm going to say that oftentimes starting about 7th grade or earlier, we call mitochondria the powerhouse of the cell. We're gonna learn to apply higher levels terms to this. So what does that mean? Well, yeah, it generates energy, but now at this level we want to say, well, how does that happen? Well, it's going to occur by breaking down food molecules, oxidizing them. When you oxidize them, you're going to take energetic electrons from the food, and you're going to be transferring them and using the energy in those electrons to power the generation of ATP. And so ATP is an energy molecule. If you're going to make it, you got to get the energy from somewhere. So where do you get it from? You're going to oxidize food, and then you're going to do that to generate or to make ATP. This kind of an answer, if we really understand what oxidizing the food, oxidizing the organic molecules like glucose, for example, and then we're going to take the energy from those electrons that we're getting from there, and we're going to use that to generate ATP. That understanding that basic idea is a higher level of understanding above powerhouse of the cell. So let's avoid using powerhouse of the cell at this level, and we'll leave that behind us for oral biology class we had in the past. So this is what I would like to see, and not only that, make sure you understand what that means. Oxidizing is something we covered back in chapter 2. So within these membranes and within the matrix, there are going to be enzymes that are within there that are going to be basically breaking down food particles, and oxygen is going to be used in this process. So this is the reason why we need to breathe in. We got to take in oxygen so that our mitochondria are able to conduct what's overall called oxidative metabolism. Now, the interesting thing about mitochondria is that they do have their own circular DNA, so they do have instructions for some of the proteins that run the metabolism within the mitochondria. And you think about that, that's a key thing there, circular DNA. What do we know has circular DNA? Bacteria, which gets us to the idea of endosymbiosis. There is plenty of evidence within the mitochondria that suggests that their origin was once an ancient bacteria. So they do have their own DNA, and that's a pretty interesting idea there. So there's a larger picture of it, and you can see within an animal cell you've got your mitochondria there. The mitochondria actually self-replicate the way bacteria do. They don't go through mitosis, they go through binary fission. Since mitosis is technically the division of the nucleus, bacteria cannot go through mitosis, they go through binary fission, and so do these mitochondria, they replicate themselves. And then chloroplasts have some of those similar features. They're going to have their own DNA. Like mitochondria, going back here, I forgot to mention they do have their own ribosomes, which are actually smaller like bacteria as well. The ribosomes that are outside of it, within the rest of the cell, they're larger, typical of eukaryotes. Same thing for chloroplasts. They have circular DNA. You can see that within the drawing there, there's a circular DNA there. They have their own ribosomes. They're going to have two membranes, an inner membrane and outer membrane. And a chloroplast is going to have other membranes where within there, some of the reactions of photosynthesis occur, the light reactions we call them. And so here I'm drawing a chloroplast with an inner membrane, outer membrane. And the way I'm going to recognize that is that we're going to have these folded membranes that almost look like stacks of quarters or coins. And each one of those little discs in there is called a thylakoid. So they got inner membrane, outer membrane like mitochondria. And we've got thylakoid, and a stack of thylakoids is going to be called a granum, and grana would be plural. Okay, so there you can see some of the terms there. The fluid-filled space that is between the inner membrane and the thylakoids is going to be called the stroma. Recall in the mitochondria just a while ago, we kind of call that fluid-filled space within the inner membrane matrix overall. So some of these features here, once you see these, this black and white drawing, whatever, it doesn't have to be green, you can recognize what it looks like. By the way, this is a real chloroplast in cross-section with electron micrograph, and then they colored it green to make it look like our ideal drawing. So this is a real observation, and this is ideal model for what a chloroplast looks like. So make sure you can recognize them and that you know some of the basic structure of it. Inner membrane, outer membrane, all of these are important in understanding when we cover photosynthesis later. The chloroplasts do look green because they're going to have a pigment called chlorophyll. And it's very important in harvesting light energy to power the process of photosynthesis. Again, you have two membranes. It's going to be key evidence, another key evidence that suggests that these came from photosynthetic bacteria that do exist. There's the thylakoids, those inner membranes, and the stacks are called grana. Grana is singular. And they do have their own DNA, and they do replicate by binary fission, just like mitochondria, just like bacteria. So the theory I was talking about, endosymbiosis, a couple of ideas here of how this might have occurred. Again, we didn't see this process. This process may have occurred in very, very ancient times, billions of years ago when cells are first evolving. But we can see that some of the first life forms were prokaryotes, right? And so the idea here is, and we see this today, we do see other cells, they're separate species, and one cell lives inside of the other one, and they have this relationship with each other. Instead of the larger cell digesting and killing the other cell, like with their lysosomes, they end up living in partnership. And over time, this partnership becomes obligatory, which means you can't separate them anymore. They become totally reliant on each other. So the basic idea here is that a proteobacterium or an ancient bacterium that uses oxygen for its metabolism got engulfed by a larger cell, a larger bacteria that just was larger and had more folded membranes, starting to look like a eukaryote. And eventually, the mitochondria starts living there and takes over the energy producing metabolism for the cell. And then later on, chloroplasts evolved from cyanobacterium. Cyanobacterium means, cyano means blue-green. These are photosynthetic bacteria. We see them today. And so some lines of these eukaryotic cells taking in cyanobacterium, they also have mitochondria. They're going to need both. And the chloroplast now starts to play the role of making, as it did for the cyanobacterium, making food for that larger relationship there. So this is the idea of how chloroplasts and bacteria came about. Endosymbiosis is an idea proposed, I believe, in the 1970s by a scientist. Her name is Lynn Margulis. And I do have an animation that is pretty interesting. It shows some of the evidence. So let's see if we can get that to run here.
The eukaryotic internal membrane system, called the endoplasmic reticulum, and the nuclear envelope may have evolved from infoldings of the plasma membrane in an ancestral prokaryotic cell. Such infoldings are common in modern prokaryotic cells. The theory of endosymbiosis proposes that a critical stage in the evolution of eukaryotic cells involved endosymbiotic relationships with prokaryotic organisms. Microorganisms that live within other cells and perform specific functions for their host cells are called endosymbionts. According to the theory, energy-producing bacteria may have been engulfed by a larger primitive cell and come to reside within it, eventually evolving into what we now know as mitochondria. Photosynthetic bacteria use photosynthetic pigments embedded in internal membranes to derive energy from sunlight. These bacteria may have come to live within early eukaryotic cells, leading to the evolution of chloroplasts. Several facts provide evidence for the endosymbiotic hypothesis. A few examples include: 1. Mitochondria and chloroplasts contain their own circular DNA, similar to DNA in bacteria. 2. Both mitochondria and chloroplasts are surrounded by two membranes. The inner membrane is probably evolved from the plasma membrane of the engulfed prokaryote, while the outer membrane is probably derived from the cell membrane of the host. 3. Mitochondria are about the same size as bacteria. 4. Mitochondria appear to have been derived from purple bacteria, and chloroplasts derived from photosynthetic bacteria.
So in this section, we're going to look at the cytoskeleton of the eukaryotic cell. And your learning objectives here are to contrast the structure and function of different fibers in the cytoskeleton. We're going to look at 3 different categories. And then you're going to illustrate the role of microtubules in intracellular transport. I have a really cool video. Hopefully it'll play toward the end of this section. So the cytoskeleton itself, what is it? It's going to be a network of protein fibers, and this is going to be typical of eukaryotic cells, plant cells, animal cells, fungi. The cytoskeleton is going to help support shape of the cell, keep organelles in fixed locations. It is dynamic itself, and that means that many of the components can be disassembled and then reassembled in different ways to fit the needs of the cell. Going back up here, the cytoskeleton can help hold some of the organelles in fixed locations, but some of the cytoskeletal elements, some of the proteins, can be used as highways for transport as well. We'll see an animation of that. So there's going to be 3 general types. You want to make sure you learn these. The thinnest or smallest diameter are the microfilaments. They're going to be made of a protein called actin. And then you're going to have microtubules. These are going to be the thickest ones, okay, in terms of their diameter. And then how are you going to remember that? The intermediate filaments. Intermediate means in between. These are going to be the middle size in diameter. Okay, so looking at the microfilaments, there's an image here, and it shows little subunits of protein called actin. And when they come together, they form fibers. So there's going to be two chains twisted up. So that's mentioned there in the description. And these actin fibers are often involved in movements of the cell or contraction of the cell. In fact, we have a lot of actin in our muscle cells, like a high amount within muscle cells. But this may be found all along, say, for example, just right along the edge of the inside of the plasma membrane. So you can see them here in the drawing here. This beige structure is supposed to represent your cell membrane, your plasma membrane. You can see those red fibers there. Those are your actin filaments. You can also see mitochondria in there, and this blue organelle looks like a rough endoplasmic reticulum right there. And this is just the other side. There's the plasma membrane on the other side. Now the microtubules, we have to remember that these are actually bigger diameters, because if you're going to be a tube, you have to be hollow inside, and that makes the microtubules thicker or more diameter than microfilaments. So there's an image right here, and you can see it does have a hollow center, and these are made of subunits called alpha and beta tubulin, and then the entire structure is called a microtubule. So you have diameters, say I'm going to circle one right there, the different color green. One is alpha and one is beta, and these subunits of tubulin come together to make your microtubules. Now, these microtubules, make a little note here, these are the most dynamic. We're going to see them mentioned, for example, when cells are dividing and we're going to pull chromosomes apart, the microtubules are involved in helping to provide highways to pull your copied chromosomes apart. Because when a cell divides, you got to make copies of your chromosomes so that each new cell gets all of the instructions, all that DNA, the genetic code. And they got to get separated. So these microtubules are going to be built up to provide highways for the chromosomes to separate to go into the two new cells. Now you can see them here. When I say dynamic, I mean that means they change. They're broken down, built back up. Okay. So again, and I mentioned how they help move things, so they do facilitate the movement of materials. And then you have your intermediate filaments, and of all of them, these are going to be the most stable instead of dynamic. So they're going to be remodeled less than the other kinds, right? And so these are of intermediate ones, and these are pretty stable, as I mentioned here, and they're really involved in helping to provide structural support for the cell. And these are going to be the ones where things like mitochondria or other things may be helped to kind of hold them into position, although mitochondria can move around a bit within there. And so here's an animation of a motor protein moving a vesicle. And we talked about vesicles, transport vesicles. And it's walking, it looks like it's walking on a microtubule. So there's your microtubule, and that is a protein. It's made of, that motor protein there, it is made of amino acids, and with the help of ATP energy molecule, the protein changes shape and quite literally walks a vesicle like it's moving along a highway. So that's a pretty cool deal. And then we have structures called centrosomes. And I mentioned this in an earlier section here. Some comes from the Greek soma, which means body. And so if we start learning the root meanings, it becomes easier to learn new words. And then central just means near the center. So the nucleus is sort of thought as the central area of the cell. These centrosomes are going to be found nearby. And so I'm going to go ahead and circle the area where the centrosome is. And in certain types of eukaryotic cells, you're going to find some structures called centrioles. And they look like this. Not all of them do. Most plant cells are not going to have centrioles, but they're still going to have a centrosome. And the centrosome, the function there is it assists in organizing microtubules, those tubular highways that we saw in the previous slide. And so they help to sort of organize them, to build them up and bring them down. So that seems to be their role there. And then you have these centrioles that are right here, and they're made of microtubules themselves. I want you to notice the arrangement there. They're arranged in triplets. So they're not very long, but they're made of 9 triplets. So we went around and we counted. There's 1, 2, 3, 4, 5, 6, 7, 8, 9, and they're triplets and hollow in the inside here. So it's referred to as microtubule triplet with 3 microtubules. So again, these centrioles are not the same thing as the centrosome. The centrioles are found within centrosomes of some animal cells and some protists, but plants and fungi tend to lack the centrioles, but they still have the centrosome, which is a region where the microtubules are arranged from. Now, it's important to pay attention to these centrioles, which are triplets, because we're going to see them as forming the basis for other structures as well, but we will see them play a role in cell division in some animal and protist cells as well. So this section is on extracellular structures and the movement of cells. And your learning objectives here are going to be to describe how cells move and then identify different cytoskeletal elements that are involved in movement. So what parts of the cytoskeleton help that movement of that cell? And then finally, we're going to look at animal cells, and we're going to look at extracellular matrix that they produce on the outside of cells. In animals, they're multicellular, and they are built on the cells organized into tissues. And oftentimes, the cells themselves secrete material on the outside that provides support and protection for those cells. So we're going to look at a cell called an amoeba. I'm going to write that down here. They're generally called amoeba, and the kind of movement that they do is called amoeboid movement. And there are other organisms besides amoebas that have this amoeboid movement. And even we have white blood cells in our bodies, part of our immune protection, that actually move in this way as well. So let's take a look at that amoeboid movement. And I want you to notice that the cell looks in the video here that was about to play looks oddly shaped, and it can actually change its shape. And those extensions from the cell have a name. They're called pseudo, which means false, and then podia, and that's something that you should know. So pod means foot, and pseudo means false. So a literal translation would be false feet. Now, IA is going to pluralize it, so false feet. So let's take a look at these false feet moving. And so there you can see those pseudopodia are moving, and those pseudopodia can allow this cell to crawl and probably not swim very well, but they can also extend those arms around food particles and take them in a process called phagocytosis. So this movement that we see here in that amoeba, again, it's called amoeboid movement, and the extensions are called pseudopodia. That movement can be tied to those actin filaments that make up microfilaments. And the protein that makes up microtubule is called tubulin. So that kind of movement that we saw there can be, depends on the cell, and the kind of movement can be tied to one or both of those types of cytoskeletal elements. And so that's what we're talking about up here in this objective. What cytoskeletal elements are involved in this kind of movement? And in this case, the microfilaments are really get involved in the cell crawling. So those microfilaments that are made of that protein called actin. And here, I have another little short video clip showing another kind of microorganism that has these little structures that extend out the cell surface called cilia. They're little hair-like structures, and they're called cilia. And cilium would be singular, so an individual cilium and then cilia. And those cilia are going to use energy from ATP, and they beat in a very coordinated fashion, and that allows the organism to swim through the water there. We also have cilia on certain cell structures, like on our respiratory tract, that will help to move mucus and any particles trapped in there. But the cilia are just small versions of flagella, and flagella is plural. So flagellum would be singular. So both flagella and the cilia are going to have this particular arrangement now, or structure. So we want to take a look here. This is going to be our plasma membrane. Okay? So that's our PM, our plasma membrane. And the plasma membrane actually surrounds, so this is supposed to be the flagella, and they cut off the rest of it. Flagella is just a much larger version of the cilia, longer. And then they cut away so we can see the internal structure. You can see it's made of those cytoskeletal elements called microtubules. Now, earlier we mentioned structures in the centrosome called centrioles. And I put a little bit of focus on this, said remember that structure, because while these are not centrioles, they have the same structure of centriole in that they have 9 triplets. This is not actually part of the flagellum or a cilium, it's at the base of them, and that structure is very much the same as the 9 triplets of a centriole, but we're going to call this a basal body when it is forming the base of a flagellum or a cilium, okay? And then you can see that once we, so again, this is going to be inside the cell, and this is outside of the cell. Okay. So now extending from there are going to be more microtubules, but they're going to have a different arrangement. Their arrangement is referred to as a 9 plus 2, and instead of triplets, it's going to be doublets, or 2 microtubules. So if we went around this here and counted the pairs, there's going to be 9 of them. Okay, so you can try that out. 1, 2, 3, 4, 5, 6, 7, 8, 9. And then you're going to have a pair in the middle. So that's where we set the 9 plus 2. Okay, and so that's the sort of that arrangement there. Now, the way this flagella moves is quite different than what we saw with the bacteria. The bacterial flagella is very simple. It's just going to have a couple of some protein fibers that don't have this complex structure in here. Now, we saw motor proteins earlier called kinesin. There's others called dynein, so there's other kinds of motor proteins. And what actually happens here is that if we were to zoom in more closely to this 9 plus 2 arrangement, and we looked at these little proteins that are sticking out from the microtubules, those are a motor protein that uses ATP to change shape. And those dynein molecules reach out to the other microtubules nearby, the other pair. And with the help of ATP, the dynein changes shape, and when it does, it pulls on the other microtubules, and that causes the entire structure, internal structure, to flex back and forth. And that's how the flagella moves for a eukaryote. And so this is a lot more complex of a structure, the flagellum itself, in terms of that extension that extends outward from the cell. And so these flagella, just like the cilia, will wave back and forth in that movement. So those microtubules become involved in movement when it comes to the waving motion of cilia and flagella. So this is just a larger image of that there. They don't have the dynein and motor proteins labeled in there, but there they are right there. And again, it's not the microtubules themselves moving, it's the dynein pulling on these flexible microtubules. It gets the whole entire structure to wave back and forth. And then some other extracellular structures we've already mentioned before. These are cell walls. We talked about them with prokaryotes. And plants, fungi, and many kinds of protists, especially the ones, the protists that we're going to study or that you'll study more closely in the Biology 2 class. There are some that are plant-like, and so they'll have a cell wall like plants made of cellulose. Now, remember, prokaryotes do have a cell wall, and it is on the outside of the plasma membrane, but it's made of peptidoglycan. That's something we covered in a prior chapter in the structure. Now, for plants and protists, which is a group of eukaryotes, and they're typically called algae, and they're photosynthetic like plants, but they're simpler. Their cell wall is going to be made of cellulose. Cellulose is that complex carbohydrate we covered in chapter 2, so make sure you know that it's made of cellulose. Fungi are going to have a cell wall, but it's made of a modified carbohydrate called chitin, and that one's going to be important to kind of remember there. Now, plants can have primary and secondary cell walls, and that's something we study more in Biology 2. Primary cell wall would be produced first, and then the secondary cell wall would be laid on the inside of it. And the reason it would be made on the inside is because the components are built inside the cell, and then they're put outside the cell and added to the cell wall there. So if we went and we took this ideal picture of a plant cell, and then there would be other plant cells adjacent to it, but we just looked at a section right here between some of the plant cells. The cytoplasm will be on one side here. So right in this area here where I'm scribbling around, that's the inside of one cell, the cytoplasm. Here's cell number 2. And then there's one out here off to the side over here. And so you can see there's two cell walls. And the middle structure there is called the middle lamella. Okay? And then you have cell wall on one side and cell wall on the other. And then right at the edge, right there, that should be where the plasma membrane is. So the plasma membrane is right on the inside there. So you're going to have primary cell wall right here, and then the thicker secondary cell wall, and then on the inside would be the cytoplasm. And again, the cell walls are made of cellulose for plants. Okay, so and then the animal cells. Animal cells do not have cell walls. Okay. And some animal cells are actually free and not really bound to any other cells or attached to any other cells, like red blood cells, for example, sperm cells. But a lot of other cells in the animal body are tied together within tissues. And instead of producing a cell wall, they produce a structure generally called extracellular matrix. And the kind of cellular matrix, extracellular matrix, depends on the kind of tissue. Like bone matrix is going to be different than cartilage, or tissues that make up tendons that attach muscles to bones and so on. So the type of matrix depends. Okay? But the matrix is basically going to be secreted by the cells, and it's going to be a mix of protein fibers and substances called glycoproteins and proteoglycans. And just in the name there, glyco means sugar, and then protein is a big part of it. So small little sugar chains of sugars, and then proteins. And then an abundant protein that's found in these intracellular matrices is a protein fiber called collagen. It's very abundant in your skin, for example. It's part of the matrix of bone and other, and so that becomes important here. So we can see there that you can see your plasma membrane right here. It's a phospholipid bilayer. Over here is inside the cell. And on the outside, extracellular means on the outside. Like instead of intracellular within, this is on the outside. And you see on the outside here, there would be a matrix, and this is your cell, and your cytoplasm is in here. But this is the outside, and there's going to be a mix of glycoproteins and other kinds of molecules. Water is going to be a component of it. And then you can see fibers like protein fibers and others called elastin and reticular fibers, different kinds of fibers. We mentioned collagen here, which is going to provide some tensile strength to there. Now, again, this extracellular matrix is going to provide support and protection for the cells, and then give those kinds of tissues their nature to it, like bone's hard, right? Cartilage is more flexible, and so on. So that's your extracellular matrix. Now, the extracellular matrix may be tied to part of the cytoskeleton inside by proteins generally called integrins. Okay, and so those are mentioned right here in the notes. And you can see how the integrin is connecting the matrix, and it specifically looks like it's connecting the collagen to some of the cytoskeleton inside. Like you can see, those are the microfilaments made of actin on the inside, or in some cases even to the intermediate filaments within there. So this table here, I would go ahead and review it. I'm not going to read through the whole thing, but it does do a comparison, and it's really good. It's like we've covered all of this. Now this is a quick summary between prokaryotes, animal, and plant cells. So cell wall here, we know prokaryotes have it, we know plants have it, made of cellulose, but animals absent, right? So I'm just going to point out a few things here. Now, we scan down here, and we start looking. Remember, prokaryotes are going to be quite simple, and they're not going to have any membrane-bound organelles. So we went and we compared endoplasmic reticulum, absent for our prokaryotes, right? And then animal and plant cells going to have them. So you can see, you can just go on down the list. Some of these structures here that are typical of eukaryotes, Golgi apparatus, membrane-bound, no nucleus, absent, absent mitochondria, chloroplast absent. After all, the mitochondria and the chloroplasts are actually almost like bacteria themselves, right? The DNA is circular for that, and so this does provide a nice overall summary. So you would want to go. You can use this as a quick review. Overall, I'm not going to read through the whole thing. These are things we've already covered. It just summarizes some of the basic characteristics. You should be able to, as a student, compare for that. The cell wall, okay, who has it? Plant, bacteria, but not animal, okay? As far as membrane-bound organelles, prokaryotes don't have it, but eukaryotes do. Plant and animal are going to have endoplasmic reticula, Golgi apparatus, and so on. So in this section, this is about how cells interact with each other. And so your learning outcomes for this are to differentiate between types of cell junctions where cells are attached to each other, and then to describe the role of surface proteins. So we're going to start by looking at these surface proteins, and these surface proteins are going to give cells sort of an identity, be able to tell one cell type from another, and so on. So cells, when they do contact each other, they're able to determine things about each other. So they sort of quote unquote read each other. And some of the molecules that are on the surface, we mentioned glycoproteins. These are going to be proteins that have short chains of sugar molecules. So we might call these not monosaccharides, not disaccharides, but modified oligosaccharides. So they're not poly, but oligo would mean few or some instead of many, like a polysaccharide would. And then there's also glycolipids. So these would be glyco again means sugar. So again, you're going to have oligosaccharides that are sticking off of lipids instead. And so all of this is on the surface of the plasma membrane. And when it comes to something like an animal that has tissues, these glycoproteins and glycolipids overall are going to have differences depending on the tissue type. So those surface markers of glycolipids and glycoproteins are going to be different when it comes to cells that make up one kind of tissue than the other. There's also a more broad category called MHC proteins, and that stands for major histo, which means tissue. Compatibility proteins. The C is for compatibility. And these generally are going to be allow your body to recognize self from non-self, from things that might invade, like a bacteria or other things get in you that you don't have these types of proteins allows your immune system to recognize, hey, this tissue or these cell types belong here, but this other thing doesn't belong here. And so the MHC stands for that big word there. But all of these things are located on the surfaces of your cells. So moving on now, from cells being able to tell each other, tell from one another who they are and if they belong there or not, we're going to now move on to cell connections, the cell junctions. Okay, and so what I have here to start with is a diagram of an organ that you find in your body. So this would be something like the small intestine, and the small intestines are tubular, so I'm going to draw like if there's the rest of the intestine, and then we just chopped into it, did a cross section. And if we did, the inside right here would be where your liquidy food would be, and as your food is being digested, it gets absorbed by the lining within there, and then will be sent off to blood vessels, which are located deeper within the organ. So this is the outside of the organ, and this is the inside of the organ. And then the wall of that organ, as you move deep from one side or the other, you're going to have different kinds of tissues, but you're also going to have blood vessels there. And so the thing about this is that we're going to want to get the nutrients that we want to. We're going to want to get them to go from where the food is in here, and we're going to want to absorb them and get them into your blood vessels so that we can circulate these nutrients to all the parts of your body that are going to need these nutrients. Okay. The reason I'm bringing this up is what we're going to do is we're going to look at a little cross section right here of that lining. It's called a mucosa in general, but it's made of a tissue called an epithelial tissue, and it's that epithelial tissue that is not only helping to digest the food, but also to absorb it and get it to the blood vessels that are deeper within the organ. So let's take a look at the diagram itself, and the diagram itself is showing some of those cells that line the inside lining of that digestive system there. And so this would be the inside right here of the intestine. Sometimes it's called the lumen. So the lumen of the intestine. And then this is the basement membrane that holds it. And then below here would be some of those other tissues where your blood vessels would be. So imagine you have your watery food up here, and it's these cells' job to pick up whatever is needed and then pass it through the cell and then transport it to the other side of the cell and then get into those blood vessels that are under here. Okay, so the idea here is we want some sort of a regulation to this. We don't want to just let anything in. And by the way, these little fringe-like structures here, those are little finger-like extensions of individual cells. They're microscopic, and they increase surface area, and they're called microvilli. Microvillus would be singular, and these were mentioned back when we were looking at the basic eukaryote structure. There was little microvilli labeled on there. Now the thing is that there are individual cells here. Here's one cell, then there's another. They have this sort of a column or cube-like shape. We don't want stuff to just sneak and slip right through there between the two cells. Okay, so we're going to have special cell junctions that prevent that. Okay, we're also going to want these cells to be connected tightly to each other so that they don't just tear apart easily. Okay, so we're going to have junctions that help do that. They help provide strength and support, structural support to just keep these tissues, because the intestines can expand, right? And when we expand, we don't want the cells to stretch apart. And the other thing we're going to need is for one cell to another to be able to communicate. And when we say communicate, we're talking about they don't have brains or anything where they can talk to each other, but at cellular level, communication is important, and it's at the molecular level, passing materials back and forth, being able to sense chemicals and electrical signals, and so on. And so we're going to look at those three kinds of structures. So what are we going to use to help keep the cells from tearing apart? Those are going to be called adhesive junctions, and typically they're also referred to as desmosomes. So this is one term I would know. The desmosome. And so what they do is they provide this mechanical support, and this is going to help keep the cells from sort of pulling apart here. And so we look more closely here. Here would be a desmosome right here, and this is an electron micrograph of a transmission microscope. So that would be your desmosome here. But let's look at the ideal drawing here between the two cells. This is one cell membrane from one cell, and this is the other cell, and this is inside that one cell, and this is inside the other cell. The desmosomes are going to have this sort of plaque-like protein here, and on the other side as well. And they're going to be connected to the intermediate filaments. Those are the ones that are the most stable. The intermediate filaments are sort of like the skeleton inside. And if we're going to keep cells from tearing apart, the plasma membrane's somewhat fluid. It's not a good structure to hold on to. You're going to want to hold on to the skeleton of each cell. And then on the middle, they're going to be these little hook-like proteins. They have their specific names. They're called cladherins, and that's all labeled in there. Don't bog yourself down in the detail of the names of these proteins, but overall, this is a desmosome, and you should know that that one's going to be there to provide this mechanical support, and it's going to attach one cytoskeleton to another. Function is to keep those cells from separating from each other completely. And then we're going to have tight junctions. And I mentioned earlier, sometimes they're called septate junctions. These are going to be the ones that are going to keep materials from just leaking through between the cells. And so really there's going to be a set of proteins that are just kind of sew tightly together the plasma membranes here. So you can see there, here is your tight junctions here, and they're just really going to hold the plasma membranes together tightly. That way this water-based fluid in here, because inside your intestine is going to be a mixture of water and digesting food. We don't want it to just slip between the cells. We got to regulate what gets in. And then we're going to have communication junctions, and the communication junctions in animal cells are going to be called gap junctions. And the gap junctions, we can see some down here, and they're basically there's the proteins that connect one cell to another, and they have these little gaps or little pores. And that's going to allow materials, chemical materials, and even electrical signals to pass directly and quickly from one cell to another. And that's very important in communication between the cells. So these are communicating junctions, and in animal cells, they're called gap junctions. So that's sort of like a vocabulary word to know. There are special proteins that have the names there. Again, don't bog yourself down in details, but those proteins are called connexons. In plant cells, those communicating junctions have a different name. They're called plasmodesmata. We're going to see that in the next slide here. So I'm going to take you back. So the plasmodesmata are like these pores that allow communication from one cell, a plant cell, to another. Now remember, the plant cells have a cell wall that's in the way there. And so those plasmodesmata are going to allow basically cytoplasm from one plant cell to the next one to communicate with each other. So we go back to the plant ideal plant cell model drawn in earlier. So this is basically a model, it's just a diagram. We're going to see some pores that actually are openings between the cell wall that allow cytoplasm and cytoplasmic materials from one plant cell to the next to be able to move materials back and forth. So let's just zoom in to that. And so we went back to that larger picture that was earlier in the chapter. Those are your plasmodesmata. Now, if I were to go and take a cross section through there, so I'll just take, say, a cross section right here, and then I look at it right here more closely, you're going to see your two cell walls. Okay, you're going to have your plasma membrane. Remember, the plasma membrane is always on the inside of the cell wall. And the middle lamella just means middle layer. That's the layer that glues two cell walls together. Well, right here is going to be one of those plasmodesmata. Okay, it's labeled right there. There's a tiny little pore, little opening in between the cell wall there and the cytoplasm, and even organelles can move or can cross through there. So here they say this shows a smooth ER kind of crossing over to the other one. But the cytoplasm itself also can exchange materials freely between there. So these are called plasmodesmata for plant cells. And they serve like similar function to gap junctions that we see in animal cells.